Thank you, Anna. I appreciate the introduction. Before getting started, I encourage everyone to review our forward-looking statements, which we've made available on our website, sigyntherapeutics.com, under the presentations button. With that said, if you're not familiar with Sigyn Therapeutics, we create blood purification therapies to perform functions beyond the reach of drugs. Our lead technology addresses life-threatening conditions for which there are no approved therapies. We call this technology Sigyn Therapy, and here's a picture of Sigyn Therapy in action in animal studies at University of Michigan. We designed Sigyn Therapy to work on the global infrastructure of dialysis machines already located in hospitals and clinics around the world. In this case, Sigyn Therapy is being delivered on a dialysis machine manufactured by Fresenius, the global leader in the dialysis industry. Our device has very expansive capabilities. It's able to address, through in vitro studies, it's been validated to address a broad spectrum of inflammatory cytokines, bacterial toxins, viral pathogens, hepatic toxins, and cytovesicles, which transport inflammatory cargoes throughout the bloodstream. Our market opportunities include some of the most significant unmet needs in global health. This includes sepsis, a leading cause of hospital deaths around the world, endotoxemia, emerging pandemic viruses, drug-resistant bacterial threats, and community-acquired pneumonia, which is a leading cause of infectious disease deaths. Our next step is an investigational device exemption that we plan on filing with FDA. This will be our first in human studies. We call this a modular study because this is a feasibility study that demonstrates safety, but at the same time, it'll allow us to bridge into pivotal studies with multiple indications. In this case, we plan on enrolling 12 to 15 dialysis-dependent end-stage renal disease patients who have endotoxemia and concurrent inflammation. There's over 550,000 ESRD patients in the United States who receive approximately 85 million dialysis treatments each year. Some things to know about this patient population: 30% of these patients die within the first year of initiating dialysis. Cardiovascular disease, followed by sepsis, are leading causes of ESRD deaths, and endotoxemia and concurrent inflammation are leading contributors for death in these indications. In North America, the market is dominated by two organizations, Fresenius Medical Care and DaVita Health. We believe that Sigyn Therapy represents a potential strategic competitive advantage to players in the dialysis industry, as it would establish potential new revenue channels through the sales of Sigyn Therapy. It provides a pathway into non-dialysis markets, and some of these markets are quite significant in size. It has the potential to reduce ESRD hospitalizations. And when these patients are hospitalized, which happens quite often, they go out of network and the dialysis companies lose revenues. And then there's potential revenue gains that result from extending the life of these companies' ESRD patient populations. So if you've looked at the value of extended life for DaVita, who has almost 200,000 ESRD patients in their network, that value's equal to $960 million for each month of extended life. Again, for each month. As it relates to Fresenius Medical Care, that value is $1.2 billion in revenues for each extended month of life of their patient population. Beyond Sigyn Therapy, we have an expansive therapeutic pipeline that's comprised of technologies to optimize drug agents, specifically chemotherapy and therapeutic antibodies. We've designed these technologies to extract circulating factors, including drug decoys, that obstruct these drugs from reaching their intended therapeutic targets. Our clinical objective is to improve therapeutic responses and reduce toxicity, but we also want to investigate the potential to take patients that are non-responders to these drugs and see if we can't turn them into responders. And that would be a life-saving feat. Chemotherapy, some things you need to know. It's the most common class of drug administered to cancer patients. Beyond toxicity is the shocking issue of poor delivery. Only 0.5% to 3.5% of a chemotherapy dose gets delivered to its target tumor site. And one of the major factors that obstruct delivery are cancer-released exosomes. These particles are highly prevalent in the bloodstream of cancer patients. And in this paper, published out of MD Anderson Cancer Center in Houston, it referenced this, that in the blood of cancer patients, it's estimated to contain about 4,000 trillion exosomes based on an average size of 100 nanometers. That's 480,000 miles of chemotherapy interference. These particles block and sequester the delivery of chemotherapy directly, but they can also export chemotherapy back out of tumor cells once it's been delivered, and they're a prime contributor to treatment resistance. Our solution, it's a system comprised of two technologies, ChemoPrep and ChemoPure. And this is associated with a patent we filed last October entitled Systems and Methods to Enhance Chemotherapy Delivery and Reduce Toxicity. The purpose of ChemoPrep is to be administered prior to the infusion of chemotherapy to sweep cancer-released exosomes out of the bloodstream to improve their delivery to their target tumor site. Our goal is to improve chemotherapy delivery with half a dose, which right up front, by that achievement, if we can achieve that, that's a 50% reduction in toxicity. We then follow up with ChemoPure, which is administered after the treatment or after the infusion of chemotherapy to remove off-target chemotherapeutic agents that didn't get delivered to the target tumor site to further reduce toxicity. And we're off to a good start. The primary means of administering chemotherapy is through nanocarriers known as liposomes. And we've demonstrated through a formulation of adsorbent components that we can be very efficient in removing these liposomes from human blood plasma. And I should also note that historically, liposomes have served as a model for exosome research based on their similar size and surface characteristics. So again, off to a good start, but we still got a lot of work to do. As it relates to therapeutic antibodies, there's 165 antibodies that generated almost 200 billion in sales last year. There's 1,200 therapeutic antibodies in the clinical pipeline. And again, limitation is poor delivery. Less than 5% of doses are reported to be delivered to the target tumor bindings or the target binding site. And in this case, anti-drug antibodies produced by the immune system in response to the antibody, soluble proteins, and again, cancer-released exosomes can express the antigen binding target of the antibody itself, and by doing so can intercept delivery. As an example, in this case, again, cancer-released exosomes. In this case, plasma levels of rituximab, which is a CD20 antibody to treat lymphoma patients. 3 hours after the infusion of rituximab, half of the drug was found to be attached to these exosomes. Our solution, we call it Immune Prep, and again, designed for use prior to the administration of these antibodies, but it's associated with a patent entitled Devices for Enhancing the Activity of Therapeutic Antibodies that we filed in May of this year. Administered prior to antibody infusion, but it incorporates a market-cleared antibody, an antibody that's already been proven to be safe and effective. It incorporates it within Immune Prep to sweep out drug decoys out of the bloodstream that would have bound to that same antibody when it was infused. And this reverse decoy mechanism, we believe, will increase the availability of antibodies to bind their intended targets. So in closing, at Sigyn Therapeutics, we create blood purification therapies to perform functions beyond the reach of drugs. Our lead technology, Sigyn Therapy, addresses life-threatening conditions for which there are no approved therapies. And in many cases, these are some of the most significant unmet needs in global health. We have an expansive pipeline of technologies to optimize the benefit of chemotherapy and therapeutic antibodies. And if we were BSF, we might say, we don't make the drugs, we make the drugs perform better. And then finally, we have a proven team with multiple first in industry achievements. The members of my team, most of the members were with me previously in a company, my last company, that we built from single shareholder startup to over 8,000 shares, 8,000 shareholders on NASDAQ, based on a single product that drove almost 200 million in market value. This product, we advanced from theoretical concept to the clinical reality of it being the first therapy to receive 2 FDA breakthrough device designations. And this technology was subsequently named a top 25 invention and top 11 medical advances in healthcare by Time Magazine. With that said, I appreciate your interest in our endeavors. In regards to contact information, I'll leave you with this slide. You can reach me at my email, jj@sigyntherapeutics.com. And again, didn't mention it at the outset, but we are publicly traded. Symbol is SIGY. Again, I thank you and appreciate your interest in our endeavors.